So welcome to the University of Connecticut Brain Imaging Research Center Speaker Tag Series. I'm the host, Fumiko Haeft, Director of BERC, the Brain Imaging Research Center, and Professor of Psychological Sciences at University of Connecticut. This speaker series is unique in that the speaker tags the next speaker. Since April, we've had Daniel Ansari, Lucina Udin, Nathan Spreng, Danny Bassett, Daphne Shohami, and today we have Roshan Cools, all fantastic cognitive or computational neuroscientists. We've had about a large number of people each session from over 100 institutions participating from all over the world, including China. I know it's hard for those people, as people from that area, as well as Europe. And today we have our first European speaker as well, so we're very excited. And as you see in this slide, you can view previous recordings from these speakers, as well as other BERC speakers, such as Tor Wager, Jim Haxby, Kim Noble, and so on. And you can also sign up to get updates on our talk series by emailing Lizzie that you see here, elizabeth.collin@uconn.edu. So some housekeeping items, I put it in the chat box here, but you will generally be muted. And I generally like, and it's often a failure, or it's always a failure, but I'm going to try it again today, to have everyone clap. I just think it's nice to hear a human clap when you're talking to the screen and before and after. So I'm going to unmute you after I introduce the speaker. And also today we're going to try this where we unmute, where you can ask questions as we go. So you feel free to unmute yourself so it's more conversational. And it seems like she has not a very long talk, so we should have plenty of time. So we'll get started. So today it's our great pleasure to introduce Professor and Dr. Roshan Cools. Professor Cools is a neuroscientist and professor of cognitive neuropsychiatry at Radboud University Medical Center and the PI of Motivational and Cognitive Control Group at the Donders Center for Cognitive Neuroimaging in Nijmegen, Netherlands. Professor Cools completed her Master of Philosophy degree, a PhD degree, and St. John's College Junior Research Fellowship, and a Royal Society Dorothy Hodgkin Research Fellowship at the University of Cambridge in Trevor Robbins' lab. Her thesis title was Parkinson's Disease as a Model of Dopamine-Dependent Frontal Striatal Cognitive Dysfunction. So I see the connection here between our previous speaker who tagged Roshan, Daphne Shohami. But I also see that she did her postdoc at the University of California, Berkeley, while I was doing my postdoc at Stanford, even though we never met in the Bay Area. She's a well-funded scientist who studies the cognitive and motivational control of human decision-making and its modulation by dopamine and serotonin. She's a member of the Royal Netherlands Academy of Arts and Sciences and the Academia Europea, and has obtained many awards, including the James McDonald Scholar Award and the Cognitive Neuroscience Society Young Investigator Award, as well as the VTMVD Awards from the Netherlands Organization for Scientific Research. She also serves as handling editor for the Journal of Neuroscience and Journal of Cognitive Neuroscience, and so on and so on. But I will stop here, so not to keep it too long, but please join me in welcoming Roshan Cools. So I'm going to unmute. I'm sorry, I forgot. Can you unmute everyone, Lizzie? Sorry. Oh, everyone's unmuted. All right. Feel free to clap. It was so quiet, I couldn't see everyone unmuted. Usually we have babies crying or dogs barking or people on the call when we unmute everyone at the same time. But we're going to mute everyone. And then we are going to unmute Roshan so she can start her talk.
Great, thank you, Fumiko. So now I can, let's see, share my screen, I think. How is this?
Yes, we can see
Is that working?
Yep.
Yeah, thanks so much, Fumiko, for inviting me, and and Daphne Shohami for nominating me for this great virtual talk series. I hear this one was going virtual already before the crisis, but I guess there's a bunch of these series now, and I guess that's a positive side effect of this horrible thing that's overcome us. I think this kind of virtual world that we're creating amongst us scientists is also a really nice illustration of how extremely flexible we are, how powerful our adaptive minds are. And I think this flexibility is actually, this flexibility with which we so rapidly adapt to these radical changes in the world around us is one of the more fascinating aspects of the human minds. And I'm really curious to see what this crisis will bring us in terms of our ability to deal with, in particular, one key dilemma that we face every day and now perhaps more than ever, namely whether to continue to exert control and take action, or rather whether to let go of control and perhaps accept our vulnerability. But let me begin by acknowledging my wonderful team before I tell you about my science, and the fact that the work that I'll present is the result of a major collaborative effort. In the first part of the talk, I'll focus on work done by the people in yellow here. Can you see my cursor? Yeah, so that's Hanneke den Ouden and primarily Martine van Schouwenburg. This is work we've done a few years ago. And then the second part of my talk will focus on work done in collaboration with Michael Frank and Andrew Westbrook. In all cases, these studies concern fairly large interventional pharmacological neuroimaging studies involving many people. So I'd like to just emphasize that, in particular, the work that I'll present today also involved key assistance from everyone here in blue. It's great to be able to work with a team like that. Okay, sorry about that. So I think it's fair to say that we live in the age of the connectome, where the brain is conceptualized as a distributed computing system consisting of multiple densely interconnected structural networks. And these can now be characterized in exquisite detail today. But the major point of my talk today is that these structural connections are relatively fixed. So an important question is, how do we adapt so flexibly to the many changes around us? The many constant changes around us. And my take-home message for my talk today is that the ascending neuromodulatory systems that originate from the midbrain, like dopamine, and I'll focus on dopamine today, might be particularly well suited to adapt the output of our structurally fixed neural systems to our constantly changing environment. So it's the chemical neuromodulators that might fulfill this function. Indeed, the human brain faces, actually any brain, not just the human brain, faces a variety of computational trade-offs or dilemmas. So depending on task demands, our networks need to exhibit stability, or rather flexibility. They need to exhibit controlled processing or automatic processing. They need to exhibit accuracy or speed. And for today, I'll focus on the first of these dilemmas, the stability versus flexibility dilemma, which is core to this construct of cognitive control, which is an ill-defined phenomenon, of course, but generally refers to that set of mental processes that allow us to obtain our goals by resisting distractions, impulses, and all kinds of temptations. It's, as you know, considered a hallmark of the human brain. We're really good at it. And so I'm sure it won't be a problem for you whatsoever to focus fully on my talk for the next 45 minutes, right? In fact,
yes,
yeah.
As long as you wish. 45 an hour, that's great.
Yeah, okay. But the question is, of course, to what degree will you actually make use of that ability, that unique ability to focus? Because focus is costly. It's fatiguing, it's tiring, but it's also opportunity costly. So many of the messages that might arrive, many of the novel inputs that might arrive on your laptop while you're listening to my talk might well turn out to be relevant. Yeah? Some messages should be processed, and they should require a form of flexibility rather than focus. And so what we need is a meta-level ability to decide when to exert cognitive control or focus, and when rather to let go of that cognitive focus and to flexibly respond to the new input. And how do we do that? That's a key question that that we ask in our group. And answering that question requires that we reconceptualize the problem of cognitive control as a problem of reinforcement learning and decision making. And my take-home message that the chemical neuromodulators, dopamine in particular, adapt the output of our neural systems to our changing environment is partly grounded in the observation that they contribute to the arbitration or decision-making between different, often opponent, computational strategies. All right, so I'll start in a bit by defining what I mean by chemical neuromodulation. And then I'll quickly zoom in on the dopamine system and highlight one of the major problems in neurology and psychiatry today. And that is that there is huge variability in both the direction and extent of dopaminergic drug effects, both across different individuals and within the same individuals across different behaviors. So some people benefit from these drugs, whereas other people are impaired by the same drugs. And next, I'll illustrate how we've begun to account for this this paradox, for these paradoxical effects, by taking you with me on a historical tour through previous pharmacological and chemical neuroimaging experiments. And that previous work really represent two relatively separate lines of work. So one focusing on striatal dopamine and its role in reward learning, reinforcement learning, and choice, and the other one focusing on prefrontal function, cognitive control, and working memory. And then in the second part of my talk, I'll present ongoing work in which we begin to integrate these dual roles of dopamine. And then I'll end by raising some important open questions for future work. Okay, so when I talk about chemical neuromodulation, I mean to refer to that set of neurochemical processes that curtail or prolong, augment or diminish effects of fast signaling in neuronal networks, often conditional on the baseline firing state of the postsynaptic cell. So this modulatory mode is often distinguished from that of fast signaling in the classic neurotransmitter mode of glutamate and GABA. It's rather the control exerted on that classic neurotransmitter mode. And I'll focus on the human dopamine system, as I said, which is so widely implicated in healthy and disordered cognitive function. And the cognitive role of dopamine is of course keenly illustrated by its implication in ADHD and Parkinson's disease, but also in various healthy states of stress and fatigue. And critically, these states are all often treated with drugs that increase dopamine. As I implied, unfortunately, there's huge variability in the direction of these effects. So there's a lot of side effects of these drugs, and they don't work in everyone at all. So consider, for example, ADHD, where symptoms of impulsivity and attention are often treated with dopamine-enhancing drugs like Ritalin. But also consider, by contrast, Parkinson's disease, where similar drugs are used to treat the movement symptoms, and they can actually contribute to impulse control disorders. So in some cases, leading to severe psychiatric abnormalities like addiction to their medication or pathological gambling. So one of the key questions that drives our work is how can the same neuromodulator dopamine have such different effects in different individuals, but sometimes even in the same individual. Okay, so let me illustrate how we've begun to address this question in the past. Until recently, I would say the dopamine community was relatively split, with some research lines focusing on striatal learning and other lines on prefrontal control. In the domain of striatal function, we've contributed early empirical foundations of the dopaminergic learning hypothesis by showing, for example, an association not only between individual differences in dopamine synthesis capacity in the striatum and performance on reward learning tasks, but also an association between these individual differences in dopamine synthesis capacity and the effect of a dopamine drug on reinforcement learning. So that low dopamine subjects, so people with low levels of dopamine, actually benefited from a dopamine drug in terms of reward learning, whereas people with high baseline levels of dopamine, you could say already optimized levels of dopamine, actually were impaired by that same drug. So dopaminergic drug effects on human reward learning depend on individual differences in baseline levels of striatal dopamine. And the implication of that is, of course, that isolation of dopamine's effects on learning and cognition requires that one takes into account the baseline state of the system. If we look at these effects across the group as a whole, we wouldn't even see any effects at all. Okay, so in the domain of prefrontal function, we've contributed to revising the hypothesis that dopamine's effects on cognition reflect primarily direct modulation of the prefrontal cortex, and have highlighted, together with other people, of course, the role of the striatum in these effects of dopamine on cognition. And the starting point of this series of work was the observation that cognitive control, or this often working memory associated cognitive control functions, are in fact multi-componential phenomena, comprising not just the stable maintenance of task-relevant representations, so this ability to focus and not be distracted, but also the, in some sense, opponent process of flexible updating of current task-relevant representations when something important changes. And based on various lines of evidence, not least the observation that there is neurochemical reciprocity between dopamine in the prefrontal cortex and dopamine in the striatum, and also work by Randy O'Reilly and Michael Frank, we put forward this hypothesis that dopamine might modulate these distinct stabilizing and flexing aspects of cognitive control by modulating distinct prefrontal and striatal regions of the brain. Oh, oops. And so that dopamine would basically enhance cognitive stability by acting on the prefrontal cortex, but that it would enhance cognitive flexibility by acting on the striatum. And so we then designed a series of studies to test that idea, where within the same paradigm we could measure this, we could obtain a measure of stability and flexibility. So we would measure distractor resistance as an index of stability and task switching within the same paradigm. For example, here in this delayed response task, subjects would be presented with faces and scenes, 2 faces and 2 scenes, and they were instructed to memorize either the faces or the scenes depending on the color of the cross. And then after a delay, in this particular example of 8 seconds, they would be presented with a distractor, which they had to ignore. And and then after the second delay, a probe would appear, and they would have to indicate whether this probe matched one of the two task-relevant images. And this kind of task provided us with a measure of task switching, because we had not only blue face trials, but also green scene trials, and people switched between attending to faces and scenes. And this generated a switch cost. People had difficulty switching between faces and scenes, but also a distractor cost, because on some trials, the distractor would be a scrambled image, and on some trials, it would be a novel image. So by subtracting those two, we could obtain a distractor cost, a measure of stability in a sense. Now, in keeping with this, this hypothesis and this functional and neurochemical opponency, we then observed in a number of studies contrasting effects of dopaminergic drugs on these two aspects, these two flexible and stable aspects of this kind of task. So this, just to illustrate that line of work, here I'm showing data from a study in patients with Parkinson's disease, tested both off their medication and on their medication. And we're measuring, I'm plotting here on the left, the switch cost, so an index of their cognitive inflexibility, higher scores representing greater difficulty with switching. And on the right, I'm showing you distractor cost from the same task, which is basically an index of cognitive instability, right? And so what we find here, and this is just one example, is that patients with Parkinson's disease, when they're withdrawn from their medication, presumably reflecting primarily striatal dopamine depletion, exhibit enhanced switch costs, so cognitive inflexibility, but at the same time, reduced distractor costs compared with control subjects. So they're less flexible, but more stable. Moreover, dopaminergic medication, when these same patients were tested on their medication, this effect was reversed, so that now on medication, they were as flexible as controls and as stable or unstable as controls. So dopaminergic medication re-established this balance between flexibility and stability. This is just one example. We've seen that multiple times also in healthy volunteers. A pharmacological imaging study that was done with a similar task demonstrated that these opposite effects on stability, or the stable and flexible aspects of this task, were accompanied by modulation of indeed distinct striatal and prefrontal regions of the brain. So just to quickly summarize, because I want to also get to the current work, is that in this case, administration of bromocriptine, this was, by the way, a study in healthy volunteers, but it's a drug that's often given to Parkinson patients. So it's a drug that increases dopamine receptor stimulation. Administration of a low dose of that drug increased striatal activity during switching. So at the time of the encoding cues, when they switched between attending to faces and attending to scenes, that's when striatal activity was modulated. Prefrontal cortex activity was left unaffected. By contrast, when we assessed distractor-related activity in the same paradigm, we found that bromocriptine left striatal activity unaffected, whereas prefrontal activity was potentiated by that same drug. So that sort of illustrates this hypothesis that dopamine might have contrasting effects on the flexible and stable aspects of cognitive control by modulating the striatum and the prefrontal cortex respectively. So cognitive control requires a dynamic adjustment of a balance between distinct brain regions depending on task demands. And dopamine might be a good candidate for implementing this task-dependent adjustment. Now, this is a line of work that is now already quite old. The next question we asked, triggered by those observations, was, well, but how then does dopamine alter the balance? You know, it's all nice and well if it does that, but how does it do that? And And here we were inspired by current gating models of striatal dopamine, according to which striatal dopamine regulates the flexible gating of actions, flexible selection of actions, you could say, by increasing activity in a go pathway of the basal ganglia while decreasing activity in a no-go pathway, the indirect no-go pathway of the basal ganglia. That's kind of actually an established older model of action control, but today it's recognized that this kind of gating role of striatal dopamine extends beyond motor actions, selection of motor actions, to the gating of cognitive tasks maintained in working memory. Again, work by Randy O'Reilly and Michael Frank and Thomas Hazy. So in essence, these gating models represent an integration of classic top-down biasing models of cognitive control with classic models of the basal ganglia, according to which striatal dopamine lowers the threshold for gating actions. And so inspired by that theoretical work, we set up a series of studies, again, empirical studies, to test the hypothesis that the biasing by the prefrontal cortex of these task-relevant processing in posterior cortex is flexibly updated only when striatal dopamine opens the gate for attentional switching, right? It's controlled by the striatum, that biasing by the prefrontal cortex of task-relevant processing is regulated by the striatum. And so in this series of studies, we presented subjects with bidimensional stimuli, like this one, face scene stimuli, consisting of overlapping faces and scenes, like this one. So on each trial, they would be presented with two stimuli like this, two of these overlapping stimuli, and they had to choose one of them. And they did that by trial and error, based on either attention to the faces or attention to the scenes. And critically, they were instructed that they had to switch attention to the currently unattended dimension, to the other dimension, as soon as they noticed or they detected that novel exemplars were introduced. So here we always combined face exemplar 1 with scene exemplar 1, let's say, and on the next trial we would reverse the pairing, and that way we could identify whether people would attend to the faces or the scenes for selecting one of the two options. And they were instructed to switch when novel exemplars in the irrelevant dimension or the currently unattended dimension were presented, were introduced. Now, these switch trials on which these novel exemplars were introduced elicited really striking signal in the basal ganglia and also in the prefrontal cortex. And moreover, not surprisingly perhaps to many, there were very strong attentional gain effects in posterior stimulus-specific regions. So bold signal in the face, fusiform face area was enhanced when people switched attention to faces, away from scenes to faces, whereas bold signal in the perihippocampal place area was enhanced when people switched to scenes. Now, the overall pattern of bold signal was nicely captured by a dynamic causal model in which striatal switch-related activity modulated effective top-down connections from the prefrontal cortex to task-relevant visual cortex. And as such, these data were consistent with the hypothesis that the striatum guides attention switching by gating the cognitive output of the prefrontal cortex. Now, I know that without giving you all the details, this might sound like magic, but let me give you a little bit more evidence convincing you that the basal ganglia is indeed relevant, plays a key role here. and so does dopamine. Because in a separate drug study, pharmacological study, we found that administration of a dopaminergic drug, again, here we used bromocriptine, so that's that same D2 receptor agonist, increased switch-related activity, cognitive switch-related activity, quite selectively in the striatum. I'm not masking anything here. This is the only effect we found. And what was interesting was that this region was located immediately adjacent to a region that exhibited a link between a behavioral index of switching on this task. So remember, people were instructed to switch when they detected novel exemplars, but not everyone did on every trial. There was a lot of individual variability. And this variability related to an index, to the fractional anisotropy index measured with diffusion tensor imaging. So an index of white matter integrity, if you want, or structural connectivity. And the point I'm making here is that this correlation with this behavioral index of switch likelihood on this task was found right adjacent to this region of the basal ganglia that was also modulated by the dopamine drug during switching in that same task.
Can I ask a quick question?
Yeah, please.
So it looks to me in detail that might be globus pallidus that's activated. And that raises the question, and I know when you're speaking to a non-basal ganglia audience, a lot of times it's easier to just say basal ganglia, but I'm a basal ganglia person. So I looked before, and some of your activation looked like it was specifically in the caudate from the image that you showed before. It was a small image, but that's what it looked like to me.
Yep.
And then this is also, of course, basal ganglia, but it looks to me like it's globus pallidus, maybe even, hard to tell, but maybe even medial globus pallidus. So how much do you see consistency across studies with the same basal ganglia region versus ventral striatum in one, caudate in another, globus pallidus in another?
Yeah, no, I think there's an interesting story there, not all of it which we have directly tested. In these tasks where what we measure is a sense of output gating. So in principle, all the information is in working memory. People know the faces and the scenes, but they kind of have to select from what's in working memory. We quite consistently see this kind of globus pallidus effect. In the kind of task switching paradigm that I've shown earlier, it was caudate, but actually most strikingly the putamen, which surprises in the early days. But subsequent work also with lesion patients that I'm not showing here confirmed actually that the putamen seems to have a really important role in this kind of task switching, which is slightly different from the paradigm I'm showing you here. So here I'm really looking at this output gating, and I think, but I haven't tested it directly, haven't compared it directly, we're doing that in a current study actually, that there might be a difference in the region, the subregion, striatal subregion, depending on whether we're looking at input and output gating.
Okay, very good, thank you, interesting.
Yeah. Okay, so this measure of fractional anisotropy indeed focused on the in the pallidum. Although I must say you were focusing on a very selective segment, and I think we don't really have that resolution with fMRI here. But we can say it's GP. What was interesting is that when we did the probabilistic diffusion tractography from this region of FA, we found that white matter fibers from these regions, run to a part of the prefrontal cortex that exhibited, and I'm showing that bundle here, that white matter tract in a different slice. So here it's this coronal slice in the bottom middle plot, and next to it I'm showing you this blue activity, and what this blue activity actually, or this blue cluster actually represents, is an effect of the dopamine drug on functional connectivity with that striatal region I'm showing in the left top panel. So interestingly, that structural region in that region in the basal ganglia or in the globus pallidus, that's sensitive to individual differences, behavioral switching, is connected structurally with a region of prefrontal cortex that also exhibits dopamine drug effects on functional connectivity measured during switching with that striatal region. So just to recap, dopamine receptor stimulation with bromocriptine modulated switch-related bold signal in a region of the striatum that connects structurally and communicates functionally with the prefrontal cortex.
Roshan?
Yeah.
Hey, thanks so much. This is really interesting. Was the were the beta weights and the globus pallidus from the bold signal correlated with the fractional anisotropy measures?
No, actually, we looked at that, but they were so it was the yeah, but yeah, we didn't find that.
Thank you.
Keep it that. Yeah. Okay. So together, these data suggest and or led us to conclude that striatal dopamine potentiates cognitive switching by gating attention to a cognitive dimension or a task dimension, a cognitive task rule, via modulating prefrontal control of task-relevant processing in sensory-specific cortex. It's a whole mouthful, I realize that. Of course, a key question remained, namely, how does striatal dopamine set the threshold for gating a cognitive task or rule? That's, in a sense, the question we started with, still unaddressed. And according to these gating models, but also according to currently popular expected value of control models, we allocate resources to a cognitive task based on its expected value. So based on the benefits versus the costs of that cognitive task. So this then leads me to our current line of work, where we integrate these dual roles of dopamine on the one hand in learning and choice, and on the other hand in cognitive control, working memory. And specifically, we've been working with this hypothesis that striatal dopamine affects cognitive control in part indirectly by modulating the value of cognitive work. And more specifically, according to the opponent-actor learning model by Anne Collins and Michael Frank, dopamine increases the sensitivity or the weight on the benefits over the costs of a cognitive task by increasing go versus no-go pathway activity in the basal ganglia. And of course, the general implication of this general hypothesis is that the cognitive effects of dopamine-related disorders, as well as their treatment, might reflect modulation of the willingness rather than the ability to exert cognitive control, or it might reflect the ability to assign value to cognitive tasks. Is probably a better way to put it. Okay, so to test this hypothesis, with Andrew Westbrook here on the top and Michael Frank, we've employed a cognitive effort discounting task that Andrew developed when he was a PhD student in Todd Bravert's lab, to assess what monetary value people assign to repeating a difficult cognitive task. And the experiment that we did in this study had 3 phases. And in the first phase, people were exposed to fairly standard working memory task, an N-back task, but then different levels. So a 1-back, a 2-back, a 3-back, and a 4-back task. So more or less difficult tasks. So they basically learned what it felt like. And in the second phase, subjects indicated by button press. So here I'm showing you eye gaze, but that's actually for the third phase, so it's a bit misleading, sorry. So in the second phase, they indicated by button press whether they preferred to repeat after the testing session the difficult or the easy version of the task in return for more or less monetary payoff. And the key point is that we titrated the monetary payoff for the easy task depending on choice, so until a sort of subjective equivalence was reached, and that allowed us to derive an indifference point which essentially corresponds to the subjective value that people assign to doing that difficult task. So just to recap, people choose between the hard and the easy task in return for money. We play around with the money, and then at some point they don't care about whether they want to do the difficult task for so much money or the easy task for a little bit less money, and then that gives us the value that people assign to that task. Of course, the key question we asked here is striatal dopamine key for this assignment of value? And so what we did here is measure striatal dopamine synthesis capacity with F-DOPA PET. And it was a large study here. So this is 100 healthy volunteers who underwent this F-DOPA PET scan. And F-DOPA is a radio tracer. When you inject it, uptake of it essentially indexes the degree to which dopamine is synthesized in the striatal terminals of midbrain dopamine neurons. And there's large variability. So some people have a high dopamine synthesis capacity, some people have low dopamine synthesis capacity. Now, here I'm plotting the value that people assign to cognitive work, the value that we estimate with that preference task that I just introduced, as a function of a dopamine synthesis capacity. Here, just median split. And I should say we did 100 subjects, but we actually gave 2 separate cognitive effort discounting tasks. So this is data from 50 subjects. So plotted here in green for the high dopamine subjects and in blue for the low dopamine subjects. And for the basal ganglia people here, this was dopamine in the caudate nucleus. And what we find is that first, subjective value declines with increasing task difficulty. So people assign a higher value to easier tasks. They prefer not to repeat doing difficult tasks. But critically, that value depends on striatal dopamine, dorsal striatal, caudate dopamine synthesis capacity. So subjects with higher levels of dopamine assign higher value to doing a difficult task. Critically, these subjects also underwent three testing sessions. Actually, we did this on three different sessions. They were also tested on placebo after intake of methylphenidate. That's, of course, the dopamine noradrenaline reuptake blocker, often used to treat ADHD, as well as a selective D2 receptor antagonist, sulpiride, to isolate the dopamine dependency of these effects. And what Andrew found was that the value of cognitive work depends not only on individual differences in striatal dopamine, but also on whether people were on drug or not. So here I'm plotting the effect of the drug for methylphenidate and sulpiride separately on subjective value. So increased scores, here are increased drug-induced increases in value, drug-induced promotion of cognitive motivation. What was interesting to us is that this effect depended on baseline levels of striatal dopamine synthesis capacity. So the effect was particularly pronounced in people with low baseline levels of striatal dopamine synthesis capacity, whereas if anything, the opposite was found, but that was not significant. But this, of course, is in line with that very early work of mine showing that drug effects can only be isolated if you take into account the baseline state of the system. Okay, now based on the opponent-actor learning model, we could actually make some more specific predictions about what those effects of dopamine should look like. Because what this model predicts is that high dopamine states are associated with increased weight on the benefits and decreased weight on the costs that people assign to these tasks. And so in the third phase of the task of this experiment, Andrew sampled choices on either end of each individual's point of indifference, which allowed us to plot the full psychometric curve here on the right, quantifying the degree to which people chose the most difficult task as a function of the difference between the subjective value of the high and low effort task that they were presented. Yeah, so on the x-axis is the difference between the subjective value of the difficult task and the subjective value of the easy task, and on the y-axis is the probability of choosing that difficult task. So of course, the greater the difference, the more likely they are to choose the difficult task. The key thing is that OPAL model simulations demonstrated that an increase in the sensitivity to the benefits and a decrease in the sensitivity to the costs of the task, of the effortful task, is associated with a steepening of the curve on the right-hand side, but a flattening of the curve on the left-hand side. And turns out that dopamine has exactly that effect. So the top panels are the same as on the previous slide. On the bottom panels show the empirical data. On the left is data from all drug sessions as a function of individual differences in caudate dopamine. The higher higher dopamine subjects exhibit a steeper curve on the right and a shallower curve on the left, suggesting higher weight on the benefits, lower weight on the costs. And we see that also as a function of drug with methylphenidate and sulpiride, increasing basically the weight on the benefits versus the costs. Now, we did this really merely to visualize the effect. Of course, they were confirmed by statistical hierarchical regression analyses, but also by dynamic gaze patterns measured during choice. And here comes these dynamic gaze patterns. And here Andrew did something clever, because what he did is he presented the benefits and basically the benefit attributes and the cost attributes of the choice separately on the screen. So, and then he measured eye gaze. So this way, he could disentangle the separate contributions of gaze at the benefits and gaze at the costs to choice. So he knew what people would choose, the left or the right option, but he could assess whether people would look more at the benefits or at the costs when making that choice. Okay, so here plotted on the y-axis of these four plots is the proportion of gaze from choice onset. At the benefits in green, I've omitted the legend, I see, but in green is the gaze at the benefits, in blue is the gaze at the costs. And I've plotted this, actually Andrew has plotted this separately for the trials on which the hard task was chosen and on trials on which the easy task was chosen. And I've plotted it separately for the high dopamine subjects and the low dopamine subjects. And what you can see is that when subjects choose the difficult task, the effect of gaze on benefits on choice was greater for high dopamine than low dopamine subjects. So they basically gazed more at the benefits when choosing the hard task than when choosing the easy task. And this difference was greater for high dopamine subjects than for low dopamine subjects. Okay, so this then together really strengthens this idea that the cognitive enhancing effect of methylphenidate and dopamine drugs reflects, at least in part, increases in striatal dopamine and associated gating of a cognitive task in proportion to their benefit-cost ratio. That they reflect a promotion of the weight on the benefits and a reduction on the weight on the costs. Maybe I can take some questions because that's complex stuff. If not, I'll just continue. I'm almost done actually. Turns out to be longer than.
I have a question about this specifically, so if you'd prefer, I can just wait.
Okay, I don't really mind.
Okay, so it's more of a conceptual question, which is, to me, one of the limitations of the concept of value is it doesn't take in, if you're paralleling value to the economic concept of value, which is what people do, this is really behavioral economics, it doesn't on its own take into account resources. In other words, if you have more financial, going to the economy, if you have more financial resources, your decision-making changes about what you value and your perception of the costs. You know, that essentially, in economics, there's value as preference and there's value as how much you're willing to pay for something. Yeah. But how much you're willing to pay for something is influenced by your financial resources. You know, the classic thing would be, my house is worth whatever somebody would pay for it. But if there was a depression, sorry to think about this, and nobody had any money, the value, quote unquote, of my house would go down. Doesn't intrinsically affect the house itself and doesn't affect its relative preference compared to other houses on my street. Just means there's a lack of resources. If if you're making a decision to buy a Maserati versus a beat up old Volkswagen, if you had more resources, you would decide one way. If you were on a grad student stipend, you would decide a different way. So this is getting, I think your results are great. I love your results. They're really interesting. I'm just getting sort of big picture back. I wonder if there's a way to add to the model by including a component that deals with resources, because cognitive resources is in fact something that you mentioned when you began your talk.
Yeah, no, it's great. And actually, I think this leads me to my next few slides, I think, because resources is one factor that's relevant, but value is context dependent in many more ways. And so that's actually the point that I want to finish with. And so maybe I can go and then we can pursue this discussion. Because, well, I'm basically visiting that if you're okay with that. I think, so what I was going to say here is that what our findings, I think, illustrate is that dopamine plays an important role in helping us arbitrate between different cognitive tasks by altering the weight on the, by altering value or the weight on the costs versus benefits. But then they do raise this obvious next question, which is if the dopamine system controls arbitration between different options or strategies or tasks, then what controls the midbrain? What controls dopamine? I think that's related to your point. Because in the case of dopamine, we can ask, how do we set the value of cognitive work? And this is actually not trivial at all, given that valuation is context dependent. For example, and I think these are other illustrations of how value is not fixed, is cognitive work, for example, is not so valuable when the environment is uncontrollable or when it is volatile or opportunity costly. So that's another set of modulatory factors. So one intriguing possibility, but now I'm sort of at the last phase of my talk where I allow myself to speculate wildly, so is that the dopaminergic midbrain is controlled by top-down control signals from, for example, medial frontal regions that compute key statistical metaparameters like environmental volatility, controllability, opportunity cost. And this fits, of course, nicely with Amitai Senhav's idea of the medial frontal cortex being important for computing the expected value of control. More speculatively, perhaps it is the medial frontal cortex that prioritizes the striatal versus frontal midbrain projections by setting their baseline tone in an environmental specific manner. From work by Antonio Strafella, we know that he has combined in a really interesting way the use of TMS over frontal cortex and dopamine PET to measure dopamine release in specific subregions of the striatum. And we know from that work that transcranial stimulation of human frontal cortex, this is in people, he's done this in people, can alter dopamine release quite selectively in subregions of the striatum that are topographically strongly connected with the stimulated region. So there's definitely room for such cortical control that's specific. And this hypothesis basically states that the cortex controls dopamine to suit task requirements. And this hypothesis doesn't fall out of thin air, but concurs generally with insights about other brain and midbrain systems, of course, the noradrenaline system, think about the adaptive gain theory, but also more recent data from the serotonin system, about the serotonin system, which was shown to switch operational mode to promote environment-specific adaptive behavior. To switch operational mode depending on characteristics of the task environment. And so this is really my last few slides, and because I think this is relevant, where we have demonstrated that methylphenidate can indeed have diametrically opposite effects on reinforcement learning depending on a key meta-parameter of the environment. So in this case, methylphenidate really optimized environment-specific adaptive behavior. And this is a study, this is data from a study by Jen Cook. She was at the time a postdoc in my lab. She's currently at Birmingham University. She tested 100 healthy volunteers on methylphenidate and placebo on a instrumental reinforcement learning paradigm where the changeability of reward contingencies varied systematically over time. It's a task designed or adapted from a task by Tim Behrens. Some of you might know his work. And critically in this task, the probability of receiving a reward for choosing the blue stimulus varies, sometimes really high, sometimes really low. But there are periods where it's stable, and there are periods where it varies a lot. And the key observation is that optimal learning rate is really low in the stable phases of the task, and it's really high in the volatile, changeable phases in the task. Essentially, people should integrate over a much longer history of outcomes in the stable phase than in the flexible phase of the task. When things change all the time, you shouldn't integrate over a long history. You should just change your behavior based on the most recent outcomes. And intriguingly, methylphenidate, Jen found, brought these learning rates closer to optimum in this task in a context-specific manner. So the drug increased learning rates in the volatile, changeable phase of the task, but it decreased learning rates in the stable phase of the task. So the implication is that the drug regulated the learning rate dynamically depending on current environmental volatility. So in future work, we might address the hypothesis that these apparently paradoxical dopaminergic drug effects reflect modulation of distinct brain regions, differentially prioritized depending on the perceived characteristics of the environment. In other words, dopaminergic drugs might alter the balance between distinct strategies by having I think I made an error there in this sentence, but by having paradoxical effects depending on the baseline tone of the system, right? And with these baseline tones being set by the environment, the perceived environment. Okay, so more generally, I think these data and these ideas illustrate how dopamine can adapt the output of structurally fixed neural systems to our changing environment. And of course, there's a whole bunch of big open questions. If we can flip the switch, the midbrain switch, who flips the switch? Can we manipulate the cortex to alter this environmental specific control of the midbrain? What is the time scale at which the midbrain switch can act? Can this switch be flipped according to state changes rather than trait differences? And a final question, which I think is really interesting, is might disordered behavior and mental illness like ADHD represent a normative adaptation to the perceived statistics of the environment? But these are open questions, and I've come to an end, and I've actually spoken for 60 minutes. What a surprise. Thank you. That's it.
Everyone's unmuted, so they need to clap.
Very good.
Thank you. So does anyone have any questions?
I have a question.
Yes, everyone else. Can you unmute yourself, Greg? Thank you.
Sure, yeah. Thanks very much for the presentation. My question is similar to John's in the sense of value perception. If it is socially determined, socially constructed, and I'm wondering what is the relationship between emotion and all of these things with attention and cognitive control, you know, because to me, I'm coming from social psychology and I know emotion is really big. So can you talk about what is your interpretation with how emotion plays in? I can't hear you.
Sorry, I have to amuse myself. Yeah,
Sorry about that.
Yeah, I think I was saying that's a really interesting question. And I've not spent that much time thinking about emotion other than the kind of treatment of emotion as changes in evaluation systems. So for example, we've done quite a bit of work on Pavlovian biases, and some people have considered emotional biases as perhaps reflecting some of these Pavlovian biases of behavior, where if we can do that, and I don't really know because I'm not really the expert in emotion, but if we consider emotional biases as reflecting Pavlovian value-related biases, then many of these effects will translate in changes in emotion, in appetitive versus aversive emotion. Also, empirically, I can confirm that, for example, depressive symptom ratings that we measure in many of our tasks, we do studies also in psychiatric patients, I haven't really presented any of those, are systematically altered by dopaminergic medication. So there are certainly mood effects. Also in these highly selected healthy volunteers, we see quite striking and consistent effects of methylphenidate on positive affect. With positive affect declining over the time of testing and staying high up on methylphenidate. Those do dissociate here from the striatal dopamine dependent change in benefits versus costs of the cognitive task. So they don't necessarily confound our effects, but I think there's something interesting here to pursue in future work. Maybe you have more specific ideas, and I'd love to hear them. Maybe at another time, but
Great, thank you so much. How can I get in touch with you?
Just send me an email.
Awesome, thank you.
Yeah.
Hi, Roshan. This is Bea Luna from the University of Pittsburgh. I see a lot of my developmental colleagues are here, including Sylvia Bungie and BJ Casey.
Yeah.
So I don't know how familiar you are with the developmental changes that have been, theorized, but also emerging evidence that has been coming up. And a lot of it speaks to these incredible findings that you're finding of low and high dopamine. We have initial evidence showing that these changes have an impact on development. So those who are showing low dopamine are the ones that are really showing the changes in cognitive development, the changes in frontal striatal connectivity that we typically see through development, which is a decrease in frontal striatal connectivity. So I think that there's important questions that this approach can answer and can also have relevance to what you posed in the beginning. Why is dopamine having different effects in ADHD versus Parkinson's? And this could also be a developmental question, as well as the emergence of psychopathology, which predominantly is emerging during the adolescent period when we're seeing changes. And I'll end with also letting you know, we had a paper recently in Nature Communications showing decreases in D2, D3 receptor availability in the striatum using PET. Something a little bit akin to synaptic pruning, while we show evidence with DTBZ that the presynaptic dopamine availability is already stable. So I'm just wondering if you have thought of these sorts of issues and how they relate to your findings.
Yeah, thanks. Very interesting. I'm unmuted, I think. I have a little bit, so not too much, but a few years ago I did some work with Evelien Krone here in the Netherlands. You might know her.
Oh, I know.
And where we've used some of these reward punishment learning tasks in cross-sectional developmental study. That was, now it's maybe, I don't know, 10 years ago or so, 8 years ago, maybe, testing this idea of this kind of adolescent peak in reward versus punishment sensitivity due to these indeed reported changes in dopamine, particularly in middle adolescence. And there was some interesting evidence actually for a nonlinear relationship there with age, although reward versus punishment sensitivity particularly was more linear. But thanks for pointing me to that work on D2 receptor availability as a function of age, because I'd love to hear more about that. That generates some very specific predictions also with this kind of cognitive effort valuation task, because there are some key questions where I guess hypothesis could go multiple ways in terms of effort and demand avoidance. With kids, for example, being very motivated, anecdotally at least. So,
yeah, I'm happy to send you that paper.
Yeah, great, thank you.
Anyone else? I know we have several more minutes, and I'm sure some people have to run, but if I may ask, I'm very curious. I know when you went into the last part of your talk, you said it was going to be kind of hand-waving or speculative, yeah. But at the very, very end, and I'm going to ask you to be more speculative, but do you have any insights or speculations about how I'm curious about the trait piece, the second one of the future or remaining questions. Do you have any kind of guesses?
Yeah, so what we see really consistently is that effects. So here I was referring to most of our evidence shows that dopaminergic drug effects vary as a function of individual differences, so trait differences as a function. Also, dopamine synthesis capacity is a trait, but we also see variation as a function of impulsivity, for example, trait impulsivity and working memory capacity. And so what I think is that for example, working memory capacity dependent changes might reflect frontal cortex mediated changes in prioritization of different dopamine projections. Now, whether this same kind of thing happens as a function of state changes is suggested by this recent work by Jen Cook and our group, showing that the drug not only has opposite effects across different individuals, or between different tasks, but even within the same task as a function of this meta parameter. So that suggests that that this system might act quite differently depending on state changes. And it would be cool to, and that's, I guess, a future study to see whether changes in cortical efficiency by stimulation, for example, alter the tracking of some kind of meta parameter, and also alter these sort of attenuate these paradoxical effects, bias the system towards having more uniform effects that are insensitive to state changes. I guess that's what I meant to imply in this.
Great. And then in the
sentence.
Cook study, the last study that you talked about, did you talk about individual differences in basic baseline dopamine levels? Or that was not part of that?
There were some of those. Yeah, no, sorry. So that, let's see. So there were also dopamine dependent. So actually, no, I should be right. This is a study that did not involve measurements of dopamine PET. So we did not have that. We do have so-called proxies of dopamine. There was some indication that methylphenidate's effects on accuracy across the task as a whole also depend on working memory capacity, individual differences in working memory capacity.
What's the proxy that you use to measure this?
So, well, we, in the current PET study, the data, I showed you Andrew Westbrook's data from the current PET study where we have 100 subjects. We included a whole range of putative proxy measures. So eye blink rate, trait impulsivity, behavioral activation scale, fun seeking kind of self-report. Also working memory capacity, listening span, it's a bit like the operation span, digit span. And we're kind of in the proxy of establishing definitively how those putative proxy measures should be combined optimally to account for most of the variability in dopamine. And what I can say now, but it's fairly preliminary, is that in fact dopamine synthesis capacity is best explained by a combination of eye blink rate and reward sensitivity fun seeking, and less so by working memory capacity. So this is partly why I think some of these working memory dependent effects of dopaminergic drugs might not reflect dependency on striatal dopamine differences, but rather sort of opponent interaction between prefrontal cortex and some striatal reinforcement system.
I was going to, thank you. I was going to ask also about how applicable it is to other types of learning. So that kind of hints at that as well, I suppose. But thank you very much.
Yeah.
Any last questions from anyone?
I wanted to follow up on the emotion question. And Roshan, you essentially, when in your answer, which I thought was good, was focusing on one dimension of emotion, which is the valent dimension of emotion. But there's a arousal dimension of emotion too, and I think that can fit in nicely with some of this allocation of cognitive resources, exertion of cognitive effort, things like that.
Yeah. I think that's a really a really good point. Let's say what shall I say about that? We started to with a postdoc of mine, Jada Sayali. We've begun to consider the role of noradrenaline actually also in cognitive effort seeking, and she's made the observation that we often talk about the cost of cognitive effort and the fact that we want to avoid it, but of course many of us seek it all the time, and there is an interesting dissociation between difficulty and engagement. So we engage with sort of intermediate levels of difficulty. There's not a linear relationship between difficulty and engagement or arousal. We're maximally aroused and engaged with intermediate levels of difficulty when we can make progress. And it's a really interesting question whether it's dopamine dependent or rather noradrenaline dependent. So I think these distinct dimensions of arousal and valence, often talked about in the context of emotion, might also depend on distinct neuromodulatory systems of dopamine and noradrenaline. I think that's a reasonable hypothesis. Yep.
Interesting. Good. Thanks.
Great. Thank you very much. So we will end this, and I think we took the full 75 minutes, so this was fantastic. Thank you so much, Roshan, for joining us today.
Thanks.
Thank you, everyone else. Thank you. Bye.
Thank you very
Bye.
much.
Nice to meet you.
Together.
Thank you.
Thanks.